Hi, beginning histology students often have difficulty on practical exams distinguishing what information is really being asked for when questions say classify or identify. So I've made this short video to help you all out. So you might ask, what is the difference between classification and identification? Let's use my picture as an example. If I ask you to identify the person in this image, your answer is Dr. Al Ghul. But if I ask you to classify the person in this image, your answer would be a female, middle-aged, brown-haired, well, I have a lot of gray as well, histology professor. So identification, to be more specific, is giving something the correct name or histological designation. That could be the name of an organ, a particular layer in a system, a region such as something like medulla or cortex. It could be the name of a specific cell type. So some other examples would be maybe organs such as spleen, the region such as medulla, a capsule or a goblet cell. Classification, however, is putting something into a series of categories that are defined by the characteristics of that thing that's being asked about. Specifically, in histology, these are typically structural or functional characteristics. For example, if you classify something by morphology, such as simple columnar epithelia, or you can classify by a functional thing like type of secretion in a mucus gland. Let's do some histological examples to make this easier for you. If you were asked to classify the tissue at the tips of the arrows, that means these cells right along this border right here, the answer would be simple squamous epithelia. You know that epithelia line surfaces and are the border between the lumen, which in this case is a blood vessel, and the underlying tissue. So that would tell you it was an epithelia. And it's very flat, and it's a single layer, so simple squamous epithelia is the answer. However, if you were asked to identify the cell type here, the answer would be very different. It would be endothelial cells. And in order to get to that answer, you would have to have had the cardiovascular system unit. We would have to have studied that system, which we have not done yet, but you will. So the questions that are asked are going to be limited by what you have already studied or have not studied. Let's do some more examples. Here's another example from our epithelium lab. If you're asked to classify by morphology the tissue indicated by red brackets, the answer would be stratified squamous epithelia. So classification requires you to differentiate the tissue from similar possibilities. In this case, maybe another stratified epithelia, stratified cuboidal or transitional, or even pseudostratified might confuse you here. All right, let's contrast that with being asked to identify the layer of the organ between these dashed lines, and that is over here. Now that includes that epithelial layer, which we just classified, and a little bit more. The answer here would be the mucosa. In order to answer that correctly, you would have to have studied the GI tract, which we haven't gotten to yet. Let's do one from the CT lab, or general connective tissue lab. If you're asked to classify by morphology the tissue in the field of view, your answer would be loose areolar connective tissue. And that classification is based on the characteristics of that tissue, which are the loose arrangement of cells and fibers and a large amount of ground substance. And all of that is in your notes. However, if you were asked to identify the structure indicated by an arrow, which is down here, it's pointing to this thin, dark line, the correct answer would be an elastic fiber. Alternatively, sometimes we put in a low mag and a high mag in the same image and ask you to identify something at higher magnification. So I could ask you to identify the circled cell, and the answer there would be a mast cell. I could have circled one in this lower mag, but that's a little bit low for cellular identification. I prefer to do that at a little higher of a magnification for you. Let's do another one. How about an exocrine gland example? If you're asked to identify the circled gland, the answer would be the lingual salivary gland. Now, in order to have answered that, you would have to have studied the oral cavity, which of course we haven't done either. And you would also have to have both a low and a high magnification to know that you were looking at the tongue. And this low magnification would tell you you were looking at the tongue, and the high magnification, you'd see these different kinds of glands here. However, if I asked you to classify the circled gland by type of secretion, then your answer would be that it's a serous gland. It would be incorrect to give me a morphological classification because I haven't asked for that. And besides which, we're not doing morphological classifications of exocrine glands because it's not really clinically relevant for you to go through all of that, knowing whether they're simple or compound or what shape they are. So it's a simple acinar gland, but you don't really have to worry about that. You just have to really be able to classify these by what they're making. So these dark red ones, full of protein, acetophilic staining, remember? And these ones would be the mucus glands. So this would be serous, and if this had been circled, it would have been mucus. So just a little review there for you. Let's do one last histological example. If you're asked to identify the region directly around the star, which is this red one here, the answer would be the medulla. In order to get to this answer, you have to go through a two-step mental process. First, you have to recognize that you're looking at the thymus. Then you have to recall that it has this outer darker region here called the cortex, and the inner lighter regions here, which is the medulla. However, if you were asked to classify by morphology the tissue in the circle here, which is in a completely different area from the first question, the answer would be unilocular adipose connective tissue. In order to get to that answer, you really would need to see a higher magnification. That's why this is shown here. And just telling me that this is adipose would not get you full credit. Probably wouldn't get you any credit because we can't really give partial credit anymore on the automated exams. So you would have to be as specific as possible and say unilocular adipose connective tissue. A lot of students forget to tell me the actual tissue. They might say unilocular, or they might say just simple stratified. If you don't tell me it's epithelium or connective tissue or muscle or nerve, whichever one of those, it's probably going to get marked wrong, because once you've learned more than one tissue type, you have to specify them on an exam in order to get full credit. So that's it, and if there's any additional questions that you have about this topic, you can always email me. I hope the presentation was useful and cleared up any confusion you may have about those terms.